Good morning. I'm Dr. Xiaoming Xu, currently the lab chief in Division of Product Quality Research within Office of Testing and Research in OPQ. In the next few minutes, I will continue the discussion of GDUFA research, but focus more on the translation of the research findings to support the development of product-specific guidances. As the title indicates, I will limit the scope of the discussion to complex products, as they are often more challenging to develop and hence more likely of needing additional research. In today's talk, I will provide an overview of the GDUFA Regulatory Science Program, as well as the Product-Specific Guidance Program. I will share some of the GDUFA research examples and specifically how they have helped fill in gaps in scientific understanding and supported the development of PSG, as well as generic review. I will end the talk with some thoughts on future challenges and opportunities for developing PSGs for complex products. The GDUFA program was first enacted in 2012, then reauthorized in 2017 under GDUFA 2, and will soon be authorized again in October this year under GDUFA 3. In addition to outlining user fees and review timelines, the GDUFA program allocates resources to the FDA to fund research to facilitate generic drug development and review. For example, since 2013, FDA has awarded 188 contracts and grants, as well as conducted numerous internal research projects led by FDA staff. This research facilitated the development of new tools and methods, which helped the FDA and industry to evaluate generic drug equivalence. A key outcome of this GDUFA research is to enable more efficient development and assessment of generic drugs, as well as improved resources for the development of PSG recommendations. A key aim of the GDUFA research is to disseminate the research findings to improve the general understanding of these products and to aid the generic industry in their product development program. Therefore, results from this research are shared through scientific meetings, as well as peer-reviewed scientific publications. The Product-Specific Guidance Program, which began in 2007, aims at facilitating the development and approval of safe, effective, and high-quality generic drug product by providing FDA's current thinking and the recommended approach for demonstrating equivalence for a specific reference-listed drug product. PSGs are published on a quarterly basis on the PSG website, which also include information related to upcoming new and revised PSGs for complex generic drug products. One of the key outcomes of the GDUFA research is the development of these PSGs. It is important to note that PSGs are FDA's current thinking, and the end applicant can propose alternative approaches that are deviating from those recommended in the PSG, but they should provide justification for the proposed approach, as well as including data and appropriate references. In addition, as negotiated in GDUFA 2, the FDA has agreed on priorities and goal dates on the development of these PSGs. All of this information can be found in the link below to the PSG website. On average, there are close to 100 NDAs approved per year that may warrant the development of a PSG. Of those newly approved NDAs, about 25% are considered to be a complex product, as defined in the GDUFA 2 commitment letter. These products include varying route of administration and dosage forms and can meet more than one definition of complexity, such as complex active ingredient, complex drug-device combination, complex route of delivery, or complex dosage forms. Two examples of complex products are shown on this slide. The one on the left is a biodegradable PLGA intracameral implant of bimatoprost, which provides intraocular pressure reduction in patients with open-angle glaucoma or ocular hypertension. Upon administration, the product is designed to provide sustained drug deliveries for several months, modulated by PLGA degradation. The complexity of this product includes complex dosage forms, as it is an implant with complex polymeric excipients, and complex route of delivery, for example, this is through intracameral delivery. Additional complexity also includes the applicator device that is used to deliver the implant. The example on the right is a double-stranded small interfering RNA indicated for the treatment of a rare disease condition called primary hyperoxaluria type 1. Oligonucleotides like this are considered complex drug substances, as they present unique analytical challenges to determine the sequence, structure chemistry, as well as impurities. These two examples highlight some of the challenges, including the need to understand the design of the drug product and come up with appropriate techniques or methods to characterize the product to show the equivalence. Product-specific guidances commonly recommend a type of study or a property of the drug product to measure. It is up to the generic applicant to select, develop, and justify the proposed approach. This includes the justification of the development methods and the evaluation criteria used to compare the generic and reference-listed drug products. As I will show in the next slide as an example, GDUFA research can help the FDA and the industry to develop and stay informed of new analytical tools for assessing a specific product or class of products. This is important because some of the properties of the complex products are interrelated and not straightforward to measure or compare. GDUFA research aims at reducing potential scientific and regulatory hurdles by providing more insight into these properties and approaches, and providing the industry with a starting point to the generic product development. Extensive research has been conducted in the area of ophthalmic emulsions, both internally and externally, to address some of the key questions facing this class of products. In this specific case, the products are complex in terms of both formulation, which is in emulsion, and the route of delivery, which is locally on the ocular surface. Two notable products in this class include cyclosporine and difluprednate ophthalmic emulsions. Both, until recently and for decades, are without a generic. GDUFA research played a critical role in the understanding of the product properties, development of the appropriate analytical methods, and helped both the development of the PSG as well as the ANDA review, which led to the final approval of the first generics for both. For example, several physicochemical properties such as particle size distribution, viscosity, drug distribution, and release have been identified as having a greater influence on the product quality and performance and may be impacted by the manufacturing process changes, and thus need more careful measurement and examination to ensure product equivalence. Further research was conducted to develop best measurement practices and evaluation criteria to support the assessment of these properties. A few research publications are listed below for your information. As you can see, GDUFA research has a significant impact not only on the ability of the FDA to develop product-specific guidances and review ANDAs, but is also a valuable resource for the generic industry use. Here, we have highlighted a few examples of recent accomplishments, both in PSG development and ANDA approvals in a few GDUFA research priority areas. These include the approval of first generic hydrocodone bitartrate abuse deterrent formulation, the first generic amphotericin B liposome injection, first generic polyparidone palmitate long-acting injectable suspension, and first generic cyclosporine ophthalmic emulsion. Ultimately, GDUFA research helped to facilitate industry generic product development and FDA's pre-ANDA and ANDA assessment. All of these findings can be found on FDA's GDUFA Science and Research website. Before ending the talk, I want to share some thoughts about challenges and opportunities in future product-specific guidance development for complex products. As we have seen with some recent NDA approvals, complexities can vary greatly between products and across disciplinary areas, and can benefit greatly from a close collaboration across multiple FDA teams. The use of new materials, technologies, and processes in these NDAs also highlight the potential scientific gaps and the need for new and reliable analytical methods, which needs new research and innovation. Furthermore, in GDUFA 3, the new goal dates on the PSG development of complex products may pose potential time constraints, especially if additional GDUFA research is needed. So early engagement of research and review will be crucial. Lastly, taking a life cycle approach towards the development of the generic necessitate better communication and collaboration across teams and with the industry. To summarize, Product-Specific Guidance Program provides FDA's current thinking on the type of studies and information to support the development and approval of safe, effective, and high-quality generic drug products. And GDUFA research plays a critical role in the generation of the evidence and knowledge and supports the timely development of the PSGs. I want to thank a few people who helped me putting together the slides. And thank you for your time and attention.